Before you begin, familiarize yourself with the Olympus Research Grade microscope and prepare it for use. First, the filter selection ring should be set to 1. Second, the 4x objective should be in alignment. Third, the fluorescence illumination shutter should be closed. Fourth, the condenser plate should be set to O for bright field. Lastly, the stage should be all the way down. The fluocell slide is a permanent mount of bovine pulmonary artery endothelial cells actually grown on the cover slip. The cells were stained with 3 different fluorescent dyes that specifically label 3 different cellular structures. Remove a slide from its container and place it under the microscope. Turn on the microscope and adjust the brightness to a comfortable level. Position the slide so the edge of the cover slip is centered in the light path. We will be using the technique of focusing on the edge of the cover slip to ensure that we are close to the correct focal plane. Using the coarse focus knob, bring the image into focus. Your image should look similar to this. Rotate the 10x objective into alignment and adjust the fine focus knob for the sharpest image. Rotate the condenser plate ring to pH1 to select the phase contrast plate for the 10x objective. Adjust the stage position and begin looking for cells. Under phase contrast, your image should look similar to this. Rotate the 40x objective into alignment. Rotate the condenser plate ring to pH2 to select the phase contrast plate for the 40x objective. Adjust the fine focus knob for the sharpest image. Under phase contrast, your image should look similar to this. You are now ready to begin your fluorescence observations. Turn off or fully reduce the illumination. Rotate the filter cube selector to 2 to select UV illumination. Open the shutter and make your observations. You may need to adjust the fine focus. Under UV excitation, you should observe fluorescence similar to this. Close the shutter and rotate the filter cube selector to 3 to select blue illumination. Open the shutter and make your observations. You may need to adjust the fine focus. Under blue excitation, you should observe fluorescence similar to this. Close the shutter and rotate the filter cube selector to 4 to select green illumination. Open the shutter and make your observations. You may need to adjust the fine focus. Under green excitation, you should observe fluorescence similar to this. Now you will capture images using the camera on the microscope and Scion image on the computer. Divert the light path from the oculars to the camera. Expose the slide to UV, blue, and green excitation. Use Scion image on the computer to capture and save images of blue, green, and red fluorescence. Your saved images will be black and white and should be similar to these. Using the 3 black and white images you've captured of the blue, green, and red fluorescence, you can create a color composite. Simply open Photoshop and press Ctrl F12 and follow the on-screen prompts. Your composite image should be similar to this.